হ্যালো এভরিওয়ান, তো কেমন আছো সবাই? আশা করি সবাই ভালো আছো। আমি সানিয়া। আজকের চ্যাপ্টার আমরা শুরু করছি সেল সাইকেল অ্যান্ড সেল ডিভিশন। সো স্টার্ট করি আমরা বেশি কথা না বলে। তার আগে অবশ্যই বলে দেবো চ্যানেলটাকে সাবস্ক্রাইব করে রাখবে যাতে তোমরা ভিডিওগুলো পেয়ে যাও, খুব তাড়াতাড়ি তোমাদের কাছে পৌঁছে যায়। সো সেল সাইকেল অ্যান্ড সেল ডিভিশন আজকে আমরা পড়বো। ইন্ট্রোডাকশন থেকে শুরু করি। Growth and reproduction are characteristic of cell of all living organisms. All cell reproduce by undergoing cell division. Division into two equal daughter cell. During the division of a cell, DNA replication and cell growth take place. Cell cycle, the sequence of events by which a cell replicate its genome, synthesized the other constituent of the cell, and then divides into two cells is called the cell cycle. An average duration of cell cycle in human cell is 24 hours, while that of yeast is 90 minutes. Phase of cell cycle. The cell cycle is divided into two phases, interphase and M phase. During M phase, the cell actually divides. The M phase start with nuclear division, karyokinesis, and ends with division of cytoplasm, cytokinesis. Interphase is the long undividing phase between two successive M phase. It was initially known as resting phase. During interphase, the cell prepares itself for cell division by undergoing both cell growth and DNA replication. It is divided into three subphases. Gap 1 or G1 phase, the cell is metabolically active and continuously grow, but does not replicate its DNA. Cell increases in size and the cell organelles except for mitochondria, chloroplast and centriole also increases in number. Synthesize or S-phase DNA replication and centriole duplication and take place without any changes in chromosome number. In animal cell, during the S-phase, DNA replication being in the nucleus and the centriole duplicate it in the cytoplasm. Gap 2 and G2 phases, protein synthesis and cell growth occurs. RNA and protein synthesis continue. Cell organelles and mitochondria and chloroplast increase in number by duplication. Some cell in adult animals do not divided heart cell, and many cell divided occasionally liver cell. These cell exit the cell cycle at G1 phase and enter into quiescent stage is where they remain metabolically active but dividing unless required. Cell division in the phase of actual cell division is M phase. M phase represented the most dramatic period of cell cycle, involving major recognition or virtually all components the cell depending on the cell type in which division is occurring. It can be mitosis or meiosis. Mitosis, it starts with the division of nucleus, karyokinesis, followed by the separation of daughter chromosome and terminate with cytoplasmic division. Cytokinesis, it is called equational division of chromosome is equally divided between two daughters. Karyokinesis, it involves the division of nucleus. Karyokinesis and nucleus can be divided through mitosis and through meiosis. Meiosis is further divided into four phases. Phases of mitosis prophase. Longest phase of division, condensation chromosome material start. In animal cell, its centriole move to opposite poles of the cell nucleus and nuclear envelope disappear. Microtubules are assembled to from meiotic spindle. Pair of chromosomes, centromere. Phase of mitosis. Phase. Arrangement of chromosome at equator and align along the metaphase split through spindle fibers. Attachment to spindle fibers to netochores and chromosome based stage of observed shape and size, number of chromosome. Spindle fibers, microtubules, centromeres of equator spindle. Anaphase, splitting of centromere, movement of chromatid towards the opposite poles. Shortest duration phase only of 2-3 minutes. That is very rapid. Chromatid are pulled apart. Telophase, chromosome condensed into long fine filaments. Spindle fibers disappear and centriole replicate. Nuclear envelope preform around the chromosome. Reappearance of nucleus, Golgi complex and ER. pair of centrioles, nucleus, chromatin threads, nuclear envelope, cytokinesis, beginning division of the cell. Cytokinesis mitosis of accomplished of the division of the parent cell into two daughter cell and by separation of cytoplasm into each daughter cell via a process called cytokinesis. Cell it occurs by appearance of a furrow in plasma's membrane. The furrow depends and join in the center, thus dividing the cell cytoplasm into two. In plant cell, due to the presence of inextensible cell wall, cytokinesis is achieved by cell plate formation. The middle lamella between two adjacent cells it begins to form in the center of the cell and grows outwards to meet the lateral walls. In some organisms, karyokinesis is not followed. Cytokinesis and its result the formation multinucleated condition called syncytium, liquid endosperm in coconut. Significance of mitosis: it is restricted to diploid cells only. However, in some plants and social insect, haploid cells are also divided by mitosis. It results in the production of diploid daughter cell with identical genetic combination, usually resulting in genetic stability. The growth of multicellular organism is due to mitosis. It also restores the nucleocytoplasmic ratio and surface volume ratio of cells. Mitosis in meristematic tissue like apical and lateral cambium result in the continuous growth of plants throughout their life. It helps in cell repair and regeneration of injured and lost body parts. It forms the basis of asexual reproduction in both plants and animals. Meiosis, it is reductional cell division which result in the production of the haploid set of chromosomes. For example, haploid gamete formed from specialist diploid cell. Diploid status of gamete is that restored at the time of fertilization. Meiosis involves two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. During meiosis, pairing of homologous chromosome and recombination between non-sister chromatid occurs so as produce four haploid cells. Meiosis 1 is subdivided into four phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 is the longest phase and it is subdivided into following five phases: liptotin, zygotin, pakitin, diplotin, diakinesis. Liptotin, compaction of chromosome; zygotin, synapsis of homologous chromosome to form bivalent formation of synaptimal complex; pakitin, bivalent, appears as tetrad crossing over, occurs between non-sister chromatid of homologous chromosome; diplotin, homologous chromosome separate except at the sites of crossover so as to form X-shaped structure, diastemata; synaptonomial complex beings to dissolve and disappear; diakinesis, terminalization of diastemata, chromosome are fully condensed, nucleus disappear, nuclear membrane breaks down. During metaphase 1, the bivalent chromosome align on the equatorial plane, and anaphase 1, the homologous chromosome separate while sister chromosome remain associated at their centromeres. Telophase 1 is marked by the appearance of nuclear membranes and nucleons. Cytokinesis followed and dyad and cell is formed. The stage between two meiotic division is called interkinesis and is generally short-lived. DNA replication does not occur during this stage. Meiosis 2 is similar to mitosis as they both involve equational division. The parental and progeny cell involved in meiosis 2 are haploid. It also involves four substages: prophase 2, metaphase 2, anaphase 2, and telophase 2. Similar to mitosis, except that these processes occur in two haploid cells. At the end of meiosis tetrad of cell, four haploid are formed. Significance of meiosis: it is mechanism of conversion of specific chromosome number of each species in sexually reproducing organism in achieved across generation. It leads to the formation of gametes, which is important for sexual reproduction. It provides chance of the appearance of new gene combination owing is crossing over. It increases the genetic viability in the population of organism from one generation to the next. Variation helps in evolution. থ্যাংক ইউ এভরিওয়ান ফর ওয়াচিং দিস ভিডিও। সাবস্ক্রাইব করে দেবে যাতে তোমাদের কাছে প্রথমেই এই ভিডিওগুলো পৌঁছে যায়। সো থ্যাংক ইউ। পরের চ্যাপ্টার খুব তাড়াতাড়ি আমি নিয়ে আসবো। আশা করি এটা তোমাদের খুব হেল্প করলো। বাই।